this all the time, but then he speaks Bengali. It's not gonna work.
No, he will speak in English. No?
I was saying Mapukije, Mr. Bengali, but you speak Bengali.
I speak in Bengali. I speak in another 4 different Indian languages and English.
I learned this statement in Hindi, but I only can say this. I don't speak Bengali. It's a lie.
It's good. It's good.
The pronunciation is okay.
Yeah. When it is not mother tongue, everybody's pronunciation sometimes a little bit differs.
And I know how to say kubvalo.
Yeah. Means it's very good. Kub means
and
very
I
good.
know how to say something that the audience knows. Agua. Agua is not Hindi, Sanjeev. Agua.
No.
What about barriga?
Bahiga. No? Bagicha is there. Bahiga is not.
A friend of mine that is from India, he told me that those words were the same, but they are not. Not even in Bengali, right?
No. Bahiga.
Okay.
Okay. Bahika, maybe from Prabahika, it's flow, probably.
Okay.
Because all the languages in India, it came from Sanskrit, so there will be some similarity.
All right. So, okay.
Vasco joined.
Hi, Vasco.
Yeah, Vasco joined.
Oi, tudo bem? Hello, Debbie. How are you?
I'm fine, brother. How are you?
I want to have a nice face, a happy face. I mean, happy, beautiful smile. I'm in my student room. Well, here. Okay. Yes, I'm in the bedroom.
Bedroom. It's okay.
It's good, It's a beautiful place, and the city is beautiful.
Okay. So
yes, the city.
Oh, nice.
Sandeep, would you like to say in Portuguese or in English that the workshop that you are organizing for this week was postponed? Are you admitting new students, maybe people in the audience? Pessoas aqui na audiência podem talvez entrar para esse curso que vai ser oferecido.
Tá bom, eu posso falar.
Okay. We will cover those things in the presentation. So in the presentation, I have so many things. So I need little bit more time.
You cover this.
I'll cover a little bit the outline of the course so that they should understand what we are teaching or what we are going to.
Okay. And before we get started, I want to advertise to the audience that there is a course for determining the COVID, the SARS-CoV, the virus subtypes using pangolin, bioinformatics tools. It's going to take place in November, but you have to register today. Anybody that's interested in to register to attend this workshop, I will place the link explaining the workshop in the chat. And you have to communicate with Tiago, with me, or with Flavia today, because we are accepting students for this workshop that's going to take place in November. I will place the information here. Anybody that still wants to register to this workshop, get in touch today. Today is the last day for signing because the professor is from UK. And we have to fill the subscription today. So Sandeep, please introduce you and tell us that you are moving as soon as possible to Bahia, to Salvador, where Vasco is from, and then please take care of the session for today, and then you do the introduction, and you be the chair for today, right? So you are in the control, Sandeep.
Yeah.
Ok, so boa tarde, gente. Boa tarde, obrigado, professor Miquel, por essa oportunidade. Tá bom que, como o professor Miguel falou, que, mas acho que melhor eu começar tipo desse, que eu vou falar um pouquinho da história, porque eu fiz meu mestrado, dissertação de mestrado com o professor Deu em Índia. Depois o professor Deu tinha uma grande colaboração com o professor Vasco. Eu cheguei aqui para fazer meu doutorado com bolsa de The World of Academic Science, TWAS. Fiz meu doutorado em 2017. Até agora estou em pós-doutorado com o professor Vasco. Já passei um concurso em Universidade Federal de Bahia. Estou esperando para todo o processamento, demorando um pouquinho. Só em algum mês eu vou mudar para o FBA. Tá bom? Obrigado, professor Vasco, professor Dev, e todos por essa oportunidade. Sobre o professor Dev, acho que vamos falar em inglês, é melhor para ele também. So, talking about Professor Dev, as I was explaining, he was my master supervisor while I was in India, and it's really, and he's, but not this Professor Dev and Professor Vasco having this collaboration since 2008, so it's a long collaboration with Professor Dev and Professor Vasco, and it's our great immense pleasure that Professor Dev is visiting professor in our bioinformatics department today, and he has more than 16 to 17 years of experience working with industries and research areas, and he will explain about these things. He has number of PhD degrees, number of master degrees, and he will explain during his presentation. So I would like to request Professor Dev and thank you to Professor Dev for this presentation. So all is yours. So if anyone has any question, they can write in the chat, and after his presentation and talk, we can discuss more about the area. Thank you, Professor Dev. Now all is yours. Thank you. Thanks a lot.
Thank you, Sandeep, and thank you, Professor Miguel, Professor Vasco, Anderson, and all the audience, all of you, very good afternoon. And today I am going to tell you about what we have done in the last 1 year in Ufmg during this COVID time. And that's why in my presentation, there will be so many things. So please be patient. So it may take some little more time. So just give me some time to explain all those things. Okay, so let's start. And if you have any problem in understanding my accent, because I will speak in Indian accent, so you can immediately stop me to ask, no problem, I can explain. Okay, let's start. Happened. So can you see my screen?
Yes, now it's okay. Yes, everything is fine.
Everything is fine. Okay, good. So today's topic is precision health in Ufmg during COVID-19. So I have arranged the slide like this. I will talk about myself, then precision health education, its global scenario, precision health in Ufmg and intitulada, then precision health in Ufmg course content, what we are teaching, And what we have done, precision health related concept, how we have applied in our COVID-19 research, some published papers we will discuss. Okay, so this is the presentation. So about myself, I am master in agriculture, then master in applied genetics. When this master in applied genetics is totally animal genetics and human, mostly human genetics. Then did the MPhil in biotechnology, that is also animal biotechnology. Then did a PhD in biotechnology, and that work was substructive genomics and reverse vaccinology. And since 2009-10, the same protocol which I developed in my PhD, that is still we are doing in our lab here in Ufmg bioinformatics with little modifications or improvements. And then did a PhD from Ufmg on bioinformatics, and that is related to cancer biomarkers and different strategy how to identify biomarkers, targets, those things in cancers. Apart from that, I have done my postdoc in bioinformatics from Ufmg under guidance of Professor Vasco, and also had a management degree postgraduate, that is in human resource management. So there are a lot of other things I am not going to so much. So experience, I have academic, industry, then I have consultancy, all those stuff. So if you see those things, I have more than 6 years academic experience, around 15 years I have the industry experience, and most of the industry are bioinformatics and personal genomics companies or precision medicine or personal. Then simultaneously I learned so many things, and 2008 onwards we started one platform, IIOB, through which we started doing the research and collaborated more than 400 500 researchers across 40 countries. Okay, and probably it was the first virtual research organization in world, probably, I don't know. In the COVID time, when we see everything is done in virtually, we started that virtual means platform for research in 2008 onwards. And for that, I got recognition from Limca Book of Records, which is means Indian version of Guinness Book, you can say. So this is in brief. Then, what are the research areas I work? Basically, I work in human biomedical and microbial genetics and genomics, biomarker and target discovery, genetic test and precision health, and last 5 years, we are working on astrobiology also, in collaboration with some labs and European space agencies and Ukraine and South Africa, like that. Okay, and this is my scientific production, more than 200 papers, 4,500 citations, 40 book chapters, 25 plus books, like that. Okay, and these are some books. I started writing or editing books in 2008. This is my first book, Genetics Fundamental Application. And then I published several omics related books, omics in biomedical or agricultural, then omics in crop science, then plant omics, then omics in bioengineering, omics in, it has 2 volumes, and Professor Vasco was the co-auditor also, then single cell omics, 2 volumes, like that pangenomics, then veterinary poultry, so all related omics, then healthcare biotechnology, then omics for personalized medicine, and this book was published in 2013 or 14. Then breast cancer, biomarkers, male breast cancer, non-invasive molecular markers in gynecological cancer, then other cancers, then precision health related books. Okay, and this is one of the very nice book for sports, exercise, and nutritional genomics, and all published by Elsevier, and artificial intelligence in precision health. Then all Povet also we published 2 books. One is this one, Bench to Bedside, and another is Omics Approaches and Technologies in COVID-19. And this book will be published this month, and Professor Vasco also co-auditor. So in brief, these are the books, and you can classify the books into various omics, then cancer and biomarkers, then last part is the personalized medicine or personalized health. Okay. Now, we have published more than 150 research articles in disease and bioinformatics, and they are related to several kinds of diseases. Wherever the scope of bioinformatics is there, I apply bioinformatics to solve the question. So these are the some, and you can see from cancer to neurodegenerative disorders like Alzheimer, then reward deficiency syndrome, addiction, all we are doing. Okay, and we have developed so many softwares also. This Axum Annotate is nothing but one of the precision health software through which you can do the variant analysis, all those stuffs. Then so many things, databases like MyRegulome. Then we have worked with also staggered disease, eye disorders from Russian countries, and so many things. So just to give a glimpse of those things. And then in COVID related work, since the COVID started, we started working with COVID, and Professor Bruno from VIA, he was the guiding force to start work on the COVID. Okay, and because initially I was not interested there, and finally we published more than 30 research papers and reviews, and some are very important papers. Okay, so like this, and this is the last paper we published, and main architect of this paper is Professor Servio from Rupreto and Professor Aristotle. And here we have shown that whatever the factors responsible for global warming, they are also responsible for the spillover of coronavirus. Okay, so this has been already highlighted in several news articles also. But the important papers which I will discuss today, this one, the multi-omics based identification of drugs and pathogenic pathogens biology, then multi-omics based prediction of symptoms, comorbid conditions in COVID and long-term consequences, then personalized COVID therapy management, how we can do. And this is very interesting, that how the SARS-CoV actually remitting lymphoma. So this is very rare case, and we tried to solve this issue with several hypotheses. So in astrobiology also, you can see at least 7-8 papers we have published, and they are highly very important in this field, because in those papers we have shown that cellulose could be the biosignature of extraterrestrial life, and that bacterial cellulose could be the key material for the colonization in the outer space, and kombucha could serve all the purposes for the space colonization. So if you are interested, you can read those papers also. So this has been already highlighted in our Ufmg news and ICB news and some other international technology network. Then in some Bengali newspaper also in India. Then this coronavirus related things also published in Indian newspaper. Then your Ufmg websites also news. And these are few only news which reported in 2020. There are a lot. And now precision health education and its global scenario. So now we will start to we are entering into the 4 areas what we are doing in Ufmg. So now precision health education global scenario, if you see, only 9 universities providing the master degree course, and that is also in precision medicine. There is a difference between precision health and precision medicine. Also, there is a difference between precision medicine and personalized medicine. So terminologies are different and the areas are also different. Now, precision medicine course, which these universities are providing, they are mostly from US and UK. And one university from from, I can say it is from Qatar, and one university from Australia. They are providing this master course. But if you see this master course, they are specifically focused on a specific area, either genome and cancer, or genomic technologies and application in some other diseases or something like that. But precision health is a little bit different. So that I will discuss later. So these are the 9 universities which are offering this master degree course in precision medicine. And in India, only one university providing the master course, and that is also, you see, artificial intelligence and precision medicine. So they are using only artificial intelligence-based computational techniques for precision medicine, and that is also related to cancers. I know their syllabus. And other 2 universities or organizations, they are providing postgraduate diploma. Okay. And that is also, you see, genomic and the personal genomic and medicine. So it's very, very focused and very particular areas. But in UFMG, what we are doing, that I will tell you. In UFMG, we started personal genome and precision health. So first, since to match with the global trend, I kept this personal genome and precision health. And we started this course in October 2021. And this is the first, probably the first course in the Latin, entire the Latin America, where the precision health related means whatever is going in all over the world, that we are discussing. And to match this global trend, we need to introduce this course in Latin America, and probably UFMG could be the best best place to start this one. Now, this is the first batch, and it is a 45 hours course. And this batch, we discussed all the aspects of precision health, and then the students also published 2 papers. So we are not only giving the students the entire scenario of what is the precision health and the certificate of attending or something, but we are giving also them the publications. The red underlined means persons or students, they are students. Okay, but all students didn't participate in the publication process. Another paper is this one. Okay. And here, this paper will be published in Inflammation. It is accepted. And here also the students participated and they did this work within this time duration. They finished the course. They also involved in the research and published those papers. In second batch, they work on a specific area of nutrigenomics, and that is also related to COVID. So this paper I will discuss a little bit because this is under review. And the third batch, the title of this course will be Biology and Informatics in Precision Health. And this is upcoming, and it will start from 12th September, and it will finish on 16th September. So within 1 week, I will finish this one. But in this case, if the students wants to do the research work, they can continue the work and they can publish the papers. Okay. So apart from this courses which I gave in UFMG as a regular course, as an extension program, I also tried to popularize this course in different other universities. So in UFTM, I gave a course lecture for I think 8 hours on precision or omics in precision health, and Professor Siomar organized this one, and we gave it virtually. Then in University of Ouro Preto, Professor Wonders, he invited me to give a lecture, and there I discussed about little bit about the course, but Professor Wonder asked me to give my experience or share my experience of industry to the students. So the 50% of this seminar was like motivation and other things, or other entrepreneurship development, or all those areas. But in the same university, we gave a 1 week means a full-time course with the name From Precision Medicine to Precision Health in the end of this August 2022, and most of the students were postdoc and or PhD students. And since they have to compulsory do a seminar or presentation, what we gave them, we asked them that what research you are doing, you just explain within 10 minutes, and next 10 minutes, what you understood from this course, how you can correlate your work with this precision health concept, or how can improve your work on the under the lens of this precision health. So that it was a very nice course, and Professor Savio, he organized this course, and we went to Prithumi and Sandeep and gave this course. Apart from that, we also I also gave this course specific aspects like sports nutrigenomics to industry professionals, and also conducted another 2 days online course in collaboration with Institute of Nutrigenomics, Ufmg, and one company from Singapore, that is Genomics, Singapore or Malaysia, I forget. So that was a very nice course also, and it was on precision nutrition. Okay, so those things we did also as an extension or to educate the people. So now precision health course content at UFMG, what we are basically the core areas we are trying to cover or give the highlights. So before that, I will give some idea about what is precision health and why it is important. Currently, if you see the most of the cases, if you see the cancers, we are finally going for a targeted therapy. Okay. So in that case, one size fits all, this concept doesn't work. Even if it is not cancer, even if it is any kind of condition, even simple headache, it won't work. For example, in this case, if I give it, suppose this population is suffering from headache and I am giving paracetamol 500 mg to all. But what will happen, I will get 4 different kinds of results. Only in one group of population, the dose and the medicine will 100% will be effective without any adverse effect. So the question is why we have different health outcomes for same condition or disease, even though we follow the suggested medication and dose, we follow suggested diet, perform common exercise, we are concerned about our lifestyle, but why this kind of difference? So if you want to know, you have to join this course to understand what is it, why it is happening. Fine. Now, if it is happening, then what I have to do, I have to go for a personalization or tailor-making. Fine. So now this tailor-making, 100% tailor-making, is it possible? Or truly tailor-made, is it possible? And if yes, how? That is also we are trying to cover in our course. Now, precision medicine and precision health, there are 2 terminologies, but these 2 terminologies now people use means interchangeably or synonymously, but there is difference. Okay, similarly, precision medicine and precision health also having difference. Now, if you want to understand the difference, you need to understand what is the aims of this precision medicine or precision health. Okay, so now when I'm talking precision or personalized medicine or whatever, it is right thing for the right person. Fine. So right thing could be your prediction of any kind of disease, prevention of any kind of disease. Okay, diagnosis, drug, dose, food, nutrition, career, well-being, any trait of the person can be means measured or can be considered. Now, if we consider this prediction, prevention, diagnosis, and drug dose for any kind of disease, that is precision medicine. Okay, but if we cover everything, okay, food, nutrition, then sports, then various other traits, even the cosmetic beauty, all those things, that will come under precision health. Okay, so therefore, precision medicine is a part of the precision health. So precision health is the basically the umbrella under which the precision medicine is a part. Fine. Now, how this personalization or precision is, or how to personalize or precise. So that also we discuss in the course. And if you see this one size fits all medicine, that is the general approach. I am giving everybody same dose, same thing, but it is not working. So we have to do the stratification, and then stratified medicine comes, where we group different population, where we group a population in different groups based on their different criteria and use the medicine. Then we do the personalization. So under that stratified group, we also use different other markers for personalized approach. And finally, we do a precision approach where we also consider environmental factors and lifestyle. So if we consider environmental factor and lifestyle in the personalized approach, that is called the precision approach or precision health medicine or precision health. Okay, and it has been started from 2015, but personalization has been started since maybe 90s or 80s. Okay, now how this personalization is done, it is basically based on the 4 P's concept: prediction, prevention, personalization, and participation. So these are the 4 P's, and how it has been done, that details we are discussing in the course. Fine. Then, as I told, lifestyle and means environmental factors. Then how this gene environmental interactions means lifestyle and your gene interacting. That is the key in your precision health concept or medicine or whatever you say. Now, our health outcome or any of these traits, mostly they are polygenic condition, and there's a complex interplay among these genes, environment, all those things. Basically, if you see a phenotype, this is basically an outcome of genetic makeup, lifestyle, and social environmental factors interactions. Okay. Now, if I quantify the interaction, then you can see the 30% of this interaction is provided by genetics, 40% is your behavior, 20% could be the social environmental factors, and 10% could be the healthcare provider, or like hospital facilities, then ambulance, or different apparatus facilities in the hospital. Now, this is the complex interplay among gene, environment, and lifestyle. Fine. And this is determining your health outcome or trait phenotype. So basically, genes are interacting with your food, lifestyle, social factors, exercise, and other environmental factors in which you are living or in which you are exposed to. So therefore, any abnormality in any of these components will be risk. Fine. And we need to identify individuals, all those factors, to identify what is the risk of that particular person. Okay. So it will be better understood maybe by this diagram. Here, if you see, this is the 20% social economic, 40% behavior, 10% biological factors, and this is the NCDs, means non-communicable disease like diabetes, obesity, all those stuff. Fine. And you see globalization, urbanization, air pollution, these are the socio-economic factors. Then behavioral factors like your alcohol use, tobacco use, all those things. Biological factors like BP, glucose level, lipid level, all those things. Then all those are basically driver force of these NCDs like CVDs, cancers, diabetes, and they are associated with different kinds of disabilities. But interestingly, if you see, all these factors, except these environmental factors, all are regulated by this 30% genetic or epigenetic risk factor, which you are inheriting or you are developing spontaneously. So to predict or to measure the risk of a particular person, we need to calculate all those factors. And how to calculate those risks, we are also discussing to some extent in this course. Now, what is the precision medicine initiative? Now, I'm not able to see this one. Okay. So precision medicine initiative was taken by Obama government in the United States in 2015, and the main objectives were the innovative approach to disease prevention and treatment that takes into account individual difference in people's gene, environment, and lifestyle, okay, to optimize and improve the healthcare. Second aspect was to give clinicians tool to better understand the complex mechanisms underlying patient's health, disease, or its specific condition. So basically, the development of different biomarkers to subclassify the disease and to understand the molecular mechanism so that preventive or the 4 P's of the precision medicine can be achieved. Okay. And third aspect is the to better predict which treatment will be most effective. And that is basically nothing but the prediction or support system. It is totally computational thing. You need to integrate the data. I'll give you the example. So here, basically, you are selecting a specific therapy for that particular person, which will be most suitable for this individual. So this is these are the key objectives. But if you see, this is 2015. But this concept, gene, environment, and lifestyle interaction, if you see, this kind of concept is already means very old. It may be 10,000 years old. And if you know about the Mahabharata and Bhagavad Gita, you can read those books. They're a nice story, and you will see so many other things in those books. There is Sanskrit means sloka, where this sloka is means told by the villain actually when he is dying, and that time the Lord asked him that you know that you did the wrong things, then why you did, and now you are dying. So the villain told, Janami dharmam nachame pravritti, janami adharmam nachame nivritti. That means I know what dharma is. Dharma means good things. Now, adharma means this adharma means bad things. So I know what is good and what is bad. But my pravritti, that means my behavior, cannot abide by. So my behavior is not helping me to not to do the wrong things. Okay. Similarly, I know the adharma means I know the wrong things, but that nivritti means my internal factor cannot revoke myself from doing it. Okay, so there is pravritti and nivritti. One is your internal factor, one is your behavior. And when you know everything, that what is good and what is not, and you are not able to do the good things because of your behavior, when you are not even stop the bad thing to do because of your internal matters, then that means that Hrishikesha means the God or the environment, that everything is the hand of the environment. Okay, so environment will decide what will be your fate. So if this is translation, or if we translate this sloka in this way, you will find it is finally nicely suiting into this precision health concept. You see here, nivritti is internal constituents, that's your gene, which is preventing you to not to do the good things. Okay, and pravritti, a lifestyle or behavior, which is allowing you to do the bad things, and then finally environmental factor also interacting, and finally you are getting the outcome. And this is the precision health concept. Okay, so now come to another part, that precision health is an interdisciplinary subject, and health itself is an interdisciplinary subject. And at least if you see, 18 subjects are required to fulfill this precision health approaches. And here you see the red ones are basically non-biological or medical subjects. You need mathematics, you need computer science, biophysics, business, economics, social sciences, engineering, all those steps. And why we need all those things, we discuss in the course in details. Now, important thing in this precision health, that omics and informatics. Without these two, you cannot do any precision health or precision medicine. Fine. Now, omics, genomics, or gene or genome, like that. So another example I am giving how our old scripts are giving us so many information or so many ideas. Okay. Now, this Om suffix is derived from Sanskrit Om. This is also Indian, very old script. It could be more than 15,000 years old. And this Om means completeness or fullness. So when we talk about gene Om means complete set of gene, and then it becomes the genome or genome. Then we can convert into genomics. We can similarly variomics, like that, and so many omics we can say. Even the exposomics are very important for precision health. Now, informatics part, there will be different categories of informatics skill sets we require, or the instruments we require, like data mining, curation, data knowledge base or databases, analytics platform, tools, software, computational technicians. Okay. Then we need biologists also, or computational biologist, and medical informatics. And how these two, omics and informatics, we need to integrate or we need to use properly for the precision health, that also we discussed in the course. Okay, it is in more details also. And data and approaches. Now, data could be big data, could be minimal data, but generally two kinds of data we need in the precision health. One is the personal data. Okay, in the personal data, there will be various omics profiles, radiology, medical records, then your exercise capacity or your metabolic profile, so many things. How many it means different profiles you will give, that much precision you will get in your things. Then one is the population data. So on respect to population, the precision means personal data will be used to identify the things. And then these two data sets will be integrated, analyzed, and interpreted, okay, through a translation mechanism where we will use bioinformatics, machine learning, NLP, or cognitive computing like IBM, Watson like things. And then we will develop this actionable personalized report and what is the recommendation. So based on this kind of approach, I will specify in the personal level what is the requirement of this person, what is the problem, and what the person needs to do. And this kind of tool is called virtual doctor or expert system, which is the one of the main aspect of the precision medicine by the US government. And that is also we discuss, and how this can be done, that is also we discuss in the course. Now, what are the current applications? Current applications are diverse. It means in a diverse areas we apply this precision health concepts. It can be your disease perspectives, it can be your personalized medicine, it can be your preventive health. Okay, and preventive health means any kind of disease, any kind of health complication. It may not be a disease, but it may be a trait. For example, if it is an acne, it is not a disease. It's simply, it's related to cosmetic. That also we can predict, and we can personal level we can identify. And other things may be the weight management, then cosmetic requirement, behavioral health, so many, any trait, whatever you want, we can come up with a precision health approach to predict, prevent, and treatment, okay, at personal level. So these are the things we cover. Now, if I say literally what we cover, this is the syllabus, but it is tailor-made based on different batches. So we basically give them idea about precision medicine, precision health, gene environmental interaction in health outcomes, Then applications of omics and biomarkers. So we focus more on this. So in precision health, genetic test and their applications. Then we also discuss about how throughput omics technology and their uses, bioinformatics tool, pipelines, databases, how it can be used in precision health, AI-based technologies also, and platforms for support system, how it can be developed and what are the under currently used. That also we discussed. And then implementation of precision health in mainly 3 or 4 areas we discussed. Disease perspectives for risk assessment, diagnosis, prognosis, pharmacogenomics in various NCDs. Then nutrition, nutrigenomics, precision nutrition, food as a drug. Then sports and fitness, how the personalized fitness can be achieved. Anybody is asking anything? Hello?
Hello. Yeah, everything is fine. You can continue.
Okay. So sports and fitness, we also saw the students when they asked that whether we can do a precision approach to identify which player will be suitable as a goalkeeper or striker. That also we can do using the precision health approach. Fine. And then several other traits, whatever trait you tell, we can mental health, addiction, intelligence, whether the person can be a good mathematician or a good poet. That also we can little bit predict using this kind of approach. Next, we discuss about designing the precision health related research, problem-oriented analysis of human transcriptome and genome or exome. Okay, and also a lot of case studies depending on the students' query. We also stored them in different courses. Apart from that, things, the precision health or precision medicine, this has a huge market potential, and it could be more than 30 to 50 billion USD in 2025 or 26. So we are also giving the idea how the entrepreneurship or what are the entrepreneurship opportunities lying under precision health, that students can also explore those areas if they are interested apart from the research. Fine. And also there will be a project and assignment as I showed you in the previous slides. Fine. So this is the outline of the course, and it is only 45 hours, so we cannot go in much detail or something like that. So I can tell it is an orientation course. Now the thing is that what is the future of this course? What could be the future of this course in Ufmg? So since I'm telling this is probably the first course in the entire Latin America, the Ufmg senior professors need to think whether we can offer this as a MSc in precision health. And if we can offer this, probably we will be the pioneer in the entire Latin America to offer this course, not as a precision medicine, but as a precision health. And if we give precision health course, so we will be the first university to offer this precision health course, not the precision medicine. So I'll request the university authorities and my senior professors to think on that if it is possible, and there is a big opportunity or prospect of this course if we introduce in Ufmg, so that we can show we can be the torch bearer in entire Latin America. So this is about the course. So let's come to the precision health concept based our research on COVID-19. So entire time we spent in COVID, during the COVID, we worked mostly on the COVID. So the precision health concept also, or medicinal approach also we used in the. So when I'm talking about the COVID, I also tell about my journey from India to Brazil, how it was so difficult to reach Brazil. Now, I came in April 2021 when the COVID cases or death rate were maximum both in India and Brazil. And if you see some picture, how difficult it was for traveling. Only Qatar Airways was that time operating, and most countries have closed their borders. So I had to use this kind of protective gear, and you see within the airlines how I have to use the shield and PPE kits. And after coming also, there are so many things. I had to undergo 5-6 days quarantine, then test, so many things. But everything was good when you have a good mentor or good friend. So my hosts were Professor Vasco and Professor Aristotle. So they made everything easy, so there was no problem. And it was very nice, but I didn't feel any problem after coming to Brazil. So it is their efforts. And also I should acknowledge the efforts of our secretaries like Saila, Fernanda, and Natalia. They helped me a lot for this travel and for other documentation. So I really thank to them. And let's move to the second part of this session. So our COVID related works, some selected works I will show here, because all those things I can't show, but if you give time, I can show 6-7 papers. So first is this one, second is this multi-omics also, okay, and third is this one, fourth is this leukemia paper, and fifth will be this minimal genome approach, and fourth, this nutrigenomics. Okay, so these papers I will try to explain in very brief way how we have used different informatics strategy to make a new protocol and easy protocol without much involvement of complex analysis, but we got around more than 80% everywhere the precision. Okay, so that is the importance. Apart from that, when I'm talking COVID, so these two books also will come. Now let's go to the first paper, nutrigenomics. This is the second batch work. Okay, and this paper is currently under review, so I won't give much disclosure, but I will tell one thing here. What is the hypothesis or concept behind this one? So here, this is the first nutrigenomics analysis for COVID. Now, what is the situation in COVID? If you consider the death rate of India and Western countries, there is a big difference. Fine. But if you consider the entire population, you won't get much difference. Entire number of or total number of death, we won't get any difference. But if you calculate per million death per country, then you will find in India, the per million death is only around 30 to 50, but in Western countries, it is more than 300 or 350. Okay, so this is one thing. Second thing, the COVID or the SARS-CoV, it's an infectious disease. So where the population density will be more, the infection rate or severity or death rate will be more. But if you consider Indian scenario, the population density per square kilometer is more than 400, whereas in European countries or in US, it is 60 to 70 maximum. Okay, so these facts are not matching with the infectious disease general principle. So why this death rate or the severity is very high in Western countries as compared to India, if I consider the death rate per million. Okay, so we, based on this hypothesis, we tried what could be the factor, and finally using transcriptome approach and nutrigenomics approach, we identified that the specific pattern of the diet and specific spices in India, which are used by Indian, they are actually preventing multiple severity pathways through which COVID actually create the severity or brings the death. But in case of Western countries, food, all the severities pathway, including your cytokines, cytokine storm, then several others, I can't without this, several others, including your carbopenia, all those pathway, they are basically activated because of this food habit and the food they use. And that is making more severity in those population and finally bringing the more death rate. So that is one, the second batch work. But this work, if you see the multi-omics-based identification of SARS-CoV infection biology drug candidate drugs against the COVID. So in this, we first identified what could be the drugs to manage the COVID symptoms, not the antiviral drug here. So we used a multi-omics approach, which is very non-conventional. Okay, so if you see this method, so we have not used different data integration strategies or hi-fi bioinformatics things. We have just used interactome, proteome, two transcriptome datasets and bibliome datasets and their combinations. And we used different gene set enrichment analysis. And here we used different strategies, basically based on the biological principle. We analyzed this data and come up with the result with more than 70% accuracy. Okay, and it was that time, it was published in probably in January or February 2021. That time there was no information about the drugs to be used for management also. So based on that time's data, we predicted 70%. But if we can consider today's scenario, it could be more than 90%, because based on the literature or reports, we calculate this accuracy. Now, in this approach, we use the consolidated biological process and pathways. So we use a consensus method basically, okay, and multi-omics consensus method. And they finally, what we can show you, different drug categories, all those things. But finally, if you see, these are the outcomes that SARS-CoV shares influenza, EBV, then measles, hepatitis, like viral infection pathways. So it is sharing different viral pathways also. And also it is sharing tuberculosis, malaria, leishmaniasis infection pathway, but they are not completely following those particular bacterial or this kind of protozoan pathway. So therefore, from this, we can identify that whether why the hydroxychloroquine or that leishmaniasis drug is not effective across all over the world. Basically, they are partially suppressing the symptoms or that particular fraction of the pathway associated symptoms when we are using those drugs, and they are not prophylaxis drug or something like that. We also identified cytokine, IFN signaling. All those are very important pathway in COVID infection. So therefore, after this report, we found one article came, if IFN mutation is there in IFN gene, then the person is more prone to develop serious COVID or all those things. Then beta-methadone in our database, dexamethasone was not there. So beta-methadone came in our, and beta-methadone became the life-saving drug in serious patients who are on ventilation, then estrogen, all those drugs. Importantly, ozone nitric acids and plasma therapy, they also came into our analysis, and these are very important for COVID management. And finally, if you see this Q-carmine, retinoic acid, vitamin D, arsenic, copper, zinc are the candidate prophylaxis agent. And most of us, we have used these components to boost our immunity. And if you see the Q-carmine, it is the product of the turmeric, which is produced mostly in India, Pakistan, or this region. Before COVID and after COVID, the export of turmeric from India, it jumped more than 2000% after COVID. So people know about the Q-carmine, how it is protecting the or boosting the immune system. So these are the things, and as I told, it is more than 70% accuracy we predicted. Second paper, which I will discuss, this is multi-omics based, highly accurate prediction of symptoms, comorbid conditions, and possible long-term complications of COVID-19. Here also, if you see, we used the same data sets, okay, which is used in the previous paper, but the analysis and the strategy of this biological function or based strategies is changed. And finally, we came up with more than 90% accuracy in the predicting the symptoms, long-term and short-term complications. Okay. So what we did, we basically classified the enriched disease into parent and child conditions. Then we predicted the long-term, short-term NCDs and infectious disease. In that category, we subdivided based on the disease symptoms, how much time it takes to develop the symptoms. So in that way, what we identified ultimately, if you see, except this dry cough and loss of taste, all the other COVID-associated mild and severe symptoms we identified using this method. Okay. Secondly, this CVDs, pulmonary, metabolic, and kidney, all came in or as a comorbid conditions in our analysis. Then myocardial infarction, then mood disorders, COPD, lung injury, all came as would be the top long-term conditions or maybe complicated conditions like that. Importantly, you see the cancers and congenital disorders. This is important, congenital disorders. Also, we found that they are associated with severity of this disease or COVID associated. But that time, the literature didn't support that congenital disease has any role or they have any interaction with COVID-19. So only 45% accuracy we got for the congenital conditions. But other cases, we got more than 90% means predictions. Then bidirectional relationship means associated disease like glioma, diabetes, psychiatric disorders, those also we identified. And based on some other hypotheses, we predict that these bidirectional conditions which are enriched, they could be the long-term consequences of the COVID along with the means other identified of our things. And currently we see that most of the cases, people are developing diabetes, so then your different mental conditions, then cardiomyopathy. And recently, yesterday, I heard a news that due to COVID, the people are getting sudden cardiac arrest, and it is very too much concern for the people. Okay, and so in this way, what I am trying to say, this we are getting 92.67% accuracy using simple strategy, but if you use proper biological interpretation of or means steps to analyze the data, you will get high accuracy. And this similar strategy can be used for other disease or other such kind of infectious disease, will be virus or bacterial disease also can be used. Okay. Now, this paper, this association of disease interaction of COVID-19 with congenital and genetic disorder. Once we found that only 45, we are getting most cases 90, 80 to 90, in some cases 100% accuracy. So why this congenital conditions is getting, we are in our analysis, we are getting only 45% accuracy. So is there anything wrong in our analysis? So what we did, we tried to review after 1 year, we tried to do a review using this first batch students that can just do a review that how many congenital or genetic disease are associated, or is there any disease interactions with COVID. And when we did that, we are surprised that this prediction is now more than 80% accuracy, okay, with the same protocol. Now, these are the students, the blue one are the students of the first batch. Now, we did this literature mining, basically the review, okay? And if you see the PubMed enrichment, the congenital heart disease, cystic fibrosis, those kind of disease are key disease. But when we plotted everything in this particular graph, you can see there are so many conditions with different kinds of interactions. Fine. This group of genetic conditions, okay, before that I should tell one thing. This is the younger or elder, or when the person is already born and getting infection. And in this case, the infection is happening during pregnancy or before pregnancy. Now, most of the cases, when we did this analysis, it was mostly the adult people when they are getting the infection. And we found this some conditions are making low or moderate form of COVID. Some are protective, like Fragile X syndrome. If somebody having Fragile X syndrome, they don't develop severe COVID. In some cases, like thalassemia or those autism spectrum, it increases the risk, but it doesn't increase the severity of the COVID-19, right? But in this particular group, sickle cell anemia or this Down syndrome, it increases risk, increases severity, as well as increases the death. So these are the case when the normal human being is getting infected. But there was no report that newborns are also showing that some abnormalities, congenital abnormalities. So in here, the autism spectrum, neurodevelopment, then congenital eye disorder, congenital tooth development, they are identified when the mother is infected during the pregnancy. So this is very important, and we need to understand or we need to find out whether COVID or this particular virus is evading this placental barrier, or it is adopting a Zika-like some kinds of mechanism, or which kind of mechanism it is considering to show its effect in newborns. So this is one thing we need to monitor, or we need to do a research on that. Long-term condition, right? So now this is the most interesting in our research. Suddenly one day I got a WhatsApp message from Professor Bruno that have you seen this one? What? No, after COVID infection or after SARS infection, this lymphoma patient is totally recovered. What you were talking? No, you see, this is the publication. Okay, then suddenly another publication we found that it is 2000. This is a Brazilian publication. Okay, 2018, the person had lymphoma, but after infection in 2021, the total remission happens for this Hodgkin lymphoma patient. Then why it is not happening to all lymphoma patients? And this is a very interesting case, and this is only for very isolated cases it is happened. It is found. So we tried to understand. Immediately we tried to focus what could be the mechanism, because in this, if we can identify that COVID or this particular, not COVID, this SARS-CoV-2, we can engineer this virus for therapeutic, or basically for cancer therapeutic purpose, like an oncolytic virus. It could be a boon for the society. So we immediately jumped into this thing, and we did a small research, and we proposed one hypothesis. I am going to tell you this one. Okay. And so what was the hypothesis, and what was about the patient's information? The patient was EBV positive, Epstein-Barr virus positive, which actually causes the lymphoma also. But there was no molecular information available for the patient. So we considered all possible genetic makeup of this patient. And in first, we hypothesized three conditions. So first hypothesis, the SARS-CoV-2 may use similar mechanism of brentuximab vedotin, which is a monoclonal antibody, and it's targeted therapy. So this is the mechanism of this particular drug, which actually targets the CD30, which is expressed in specific lymphoma, Hodgkin lymphoma cases. And this monoclonal antibody actually carries this MMAE, which upon binding to this CD30, it releases inside the lymphoma cell, and this MMAE blocks the spindle formation, and therefore it brings the apoptosis or cell death or cell cycle arrest. So this is mechanism. So we first hypothesis we consider this could be a mechanism. So in this case, what could be? So SARS-CoV will bind to the cell surface of this lymphoma using some CD molecules, and then it will get inside the cell and release some of its protein, and which will act like MMAE and kill the particular lymphoma cell. So this is the first hypothesis. Second hypothesis, so for that you need to understand how this microtubule complex is formed and how and where the blockage can be possible. So this is one thing which will require to do the analysis. The second hypothesis, as the patient was EBV positive, some SARS-CoV protein may interact with the LMP1 of the EBV, because EBV, once it is infected, it releases this LMP1 protein, and this protein integrates into cell membrane, and its cytoplasmic domain actually having specific residues which triggers upon some phosphorylation, triggers the kappa beta pathway. So these two domains, whether these two domains are blocked by any SARS-CoV protein. Okay. Then third hypothesis, we presume that any protein of the SARS-CoV may bind to PD-1. So this death receptor signaling also means essential, or not essential, but in some cases, this PD-1 positive lymphoma, we use the PD-1 targeting drugs to kill the cell or to stop this PD-1 signaling for killing the lymphoma cell. So these three hypotheses were we considered, and we started working on that. What we did, we simply used these strategies. Basically, it is a structural biology work, and most of the work Dr. Sandeep Tiwari, he did the dockings and did the analysis and all those stuffs. So we took all the proteins of SARS-CoV, and wherever it is not crystal structure, we made the, what it is called, we made the 3D structures. And we also consider whatever CD means possible in the lymphoma. So all CD we took, and also we took the gamma tubulin, PD-1, those things, and also we modeled this LMP1 from the EBV. And then we did the protein-protein docking, analysis of the crystal structures and dock complex, and then residue mapping, specific where it is binding and whether there is overlapping, all those things we did. And we come up with this kind of things. See, the spike may bind to 4 CD molecules. Okay. 15, 27, 44. So there is a possibility they can bind in these, or that spike RBD can bind in those particular CD molecules. And these are the different residues. And these residues also the RBD binding domain. And then we've identified that AM and ORF3 protein of this gamma tube of this particular SARS-CoV, they can hinder this gamma tubulin complex and tubulin nucleation. So if you see the structure of the gamma tubulin, or this what I can say, the tubulin structure, that GCP1 and GCP3, 2 and GCP3, they bind laterally, and on top they have this gamma tubulin. And these proteins, ORF3A, it is binding basically this lateral position of this GCP2 and GCP3, and probably it means inhibiting this lateral binding of GCP2 and GCP3. So this we identified or predicted. Then we also predicted that both the AM and ORF3 proteins may interact with GCP at its tube binding site. So here, if you see this blue, this green is the AM, and this is the tube or tubulin, and this is GCP3. So it is basically this AM is binding the same residues which the gamma tubulin uses to bind to GCP3. So there is a match. We didn't do the simulation because it was impossible to do so many docked complex, more than 200, 300 complex we created, and it was not possible. So in this way, we found that also we hypothesized, this is the final structure if you see. This SARS-CoV probably can bind through its spike, CD15 or CD27 or CD45 or CD152. And once it binds, then it may go inside the lymphoma cell, and then it releases its proteins. And in GCP2, this lateral position M protein binds, and GCP3 ORF3 binds. So therefore, this complex formation doesn't take place, and this microtubule nucleation may stop. And the second case is the SARS-CoV may directly bring the apoptosis, which already reported in one paper by other group, that it can directly bring the apoptosis. So it was not in our hypothesis. Our second hypothesis is that TubeG1 and GCP3 binding location, ORF3 is binding, ORF3A. So this TubeG1 and GCP3 binding also stop, and therefore this complex will not form. And then if this complex is not formed, this complex will not form, this GCP3, GCP2 and TubeG complex will not form, and then cell cycle will stop. The second hypothesis that EBV, we found this TARD and this other domain, these proteins, MPRO, spike, NSP7, NSP10 may bind. And if they bind in this, that NF-kappa beta signaling, that oncogenic signaling pathway will stop, and then its cell will undergo apoptosis or growth arrest. We also found that PD signaling, PD1 signaling may also stop by spike binding, because PD has a possibility to bind with the spike. So these are the hypotheses we provided, and it's published in Viruses. And now we are writing a project to do this, what I can say, experimental validation of these works. Okay. So this is one work. The next work is the personalized COVID management. Here we did the same thing. We used different omics data, and here we basically used the pathway subtraction or Venn approach, and then we identified the common pathways for different comorbid conditions and COVID, and combined comorbid conditions and COVID, and based on that, those particular common pathway, we identified the drug targets and identified the targeted drugs from FDA approved drug list. So here you can see for the diabetes, this is the Venn approach, this is the common genes. Then we did the network analysis to identify which is the hub gene and key, and then we identified the drugs. So in this way, we did for hypertension, we did for cancer, we did for chronic kidney disease, like that. And these are the different common pathways. And if you see this table, shared pathway based targets, here you can see single, suppose cancer, these are the pathways. But when there is a diabetes plus cancer plus COVID, this is the target. Similarly, if, for example, the person is having diabetes, heart disease, and also chronic kidney disease, then MAPK1 will be the target, and the targeting drug would be this one. So in that way, the pathway, common pathway-based targets, and also hub gene-based targets and targeting drug also we identified. And the interferon therapy is, before when we submitted this article, we published it in preprint. And after publication in preprint, we found that Indian government, that DGCA, or I forget, this drug authority who gives the permission to use the drug. So they gave the interferon therapy for the COVID patients who are very severe. But that was also after this publication. But I don't know whether they found this paper and then they recommended it. Don't know. But in our analysis or our results, what we can say, if the COVID patient, or if the COVID patient having the condition that is suffering from chronic kidney disease, then the IFN therapy will be most effective. It's not for all cases. So similarly, different combinations of comorbid conditions and their, if these conditions are infected with COVID, then what could be the targets we identified. Fine. Now, this is an important also work, and this is the first batch did, and the hypothesis is, and this hypothesis also, Professor, or not hypothesis, the question. This is my last presentation, so just give me 5 minutes to explain this one. Here, one day we were discussing all those things, and Professor Bruno and Professor Aristotle, they told Dave, you know, now this Omicron is infecting more people, and kids are also getting infected. Okay, so why it is happening? Can we do some research on that? So I just thought that how it can be, what we can do all those things, because now so many genomes, so many technology are used, then how we can do. So basically the question was, why Omicron shows hypertransmissibility with decreased disease severity? That is the question. And here we used a minimal number of genome-based approach. We used only 4 genomes, okay? And based on that, we identified why Omicron show transmissibility and less disease severity, okay? And we got several comments from the reviewers that when you have millions of genome, where you have so much robust technology available, why you are limiting this within minimal genome or something like that? Then we told that if I don't have in my lab a supercomputer, I can't go for a simulation for such kind of number of complexes, then how I can use or how I can do the research? So I have to do a research with minimal resources and maximum output or maximum precision. So that strategy or that idea, or I think always in the same direction, that minimal investment, maximum output, because I spend maximum time in the industry. So this is the industry principle. So that's why the minimum effort, and if with that minimum effort, we can give the explanation why it is happening. So in this method, we used some principles of reverse vaccinology and structural biology, that much. We didn't do any robust complex analysis or simulation. So these are the 4 genomes we took. One, that is the RepSec, and we selected randomly 1 gamma, 1 delta, and one Omicron from the gene bank. And that is also deposited at the end of the December, last quarter of the December. Okay, and what the analysis we did the mutation mapping, prediction of pathogenic antigenic property, prediction of cytokine. So all these are in the vaccinology approach. Then identification of immune-associated human protein that interacts with ORF8, M, N, and other SARS-CoV proteins, because we found some literature that these proteins are associated with such kind of severity or something like that. That then protein 3D structure, protein docking, binding strength calculation, okay, for mutated proteins, and analysis of the effect of the mutation in protein-protein interaction. So these things we do, simple analysis, okay. And we've identified, if you see, this is the thing, the pathogenicity order actually decreased from Wuhan to Omicron. So Omicron having the less pathogenicity. Okay, that's why it is not creating severe disease. Antigenic property is highest in Omicron, and that is also, if you see the RBD of this Omicron, this is the showing highest antigenic property. So if the vaccine is developed using this Omicron RBD, that will be more effective probably. Okay. Then we also found that pro-inflammatory cytokine production is lowest in Omicron. IL-6 production also lowest in Omicron. So, but IFN gamma and IFN IL-4 production in Omicron is highest. Okay, so what is the hypothesis? Decreased disease severity by Omicron variant may be due to its decreased pro-inflammatory and IL-6 simulation and increased IL-4 and interferon gamma induction efficacy. So this is one, probably the mechanism. And second, or next mechanism, mutation in the N protein are associated with a decreased IL-6 induction and human DTX21 mediated increased IL-4 production for the Omicron. So this is the molecular mechanism, why this IL-6 is produced less and IL-4 is produced more in Omicron. Okay. Due to the mutations, stability of all these proteins like ASM, N, and E proteins are decreased in Omicron as compared to others. So once the protein stability is decreased, then there will be so many structural problems in the virus and its survivability. So what is happening, although stronger, we found that, and others also reported that Omicron spike RBD binds more strongly to ACE2, but due to its lowest, that spike RBD or spike stability, the spike RBD interaction is weak for the systemic infection for causing severe disease. Okay, so this is one important finding from this. And finally, highest instability of Omicron E protein, okay, may be associated with the decreased viral maturation, because the envelope protein is not stable, then maturation also will not be more. Okay, and if viral maturation is less, then viral load will be less. So leading to, so that is actually leading to less severe disease and also faster recovery in case of Omicron infection. So this is a very simple analysis, but at least it is explaining why this kind of things is found, or specifically this pathogenicity is found in case of the Omicron and not found in other variants. So these are the papers we discussed, which are maybe related to precision health and produced by our students. And now let's, who are my key collaborators in Brazil? There are a lot of people, but I can't show them in a single slide. So Professor Vasco, Professor Aristotle, Professor Savio, and Bruno, they are my key collaborator here. And there are a lot of my friends and so many people here. They are my collaborator, friend, or nice partner also. Partner in the sense means research partners. And apart from that, I work with more than 450 people from 40-50 countries. I don't know how many they contribute in my books, in my research, in my reviews. Okay, also I do some research with them. So this is in a brief, for I can finish this one. So let's have, if you have any question, we can discuss. Yeah.
Sure.
Questions?
Any question?
Yeah, people in audience can ask the questions here, opening the camera and microphone. There are some people in YouTube. If they would like to post the questions there, we can bring the questions here. So, okay. Debbie, nice talk. It was great. I was wondering, the data that you use, you depend on for analysis, how much of data would be okay if you had information from exome, only for the exomes? I'm asking this because I would like to post another question, is if you could imagine a project depending on novel sequencing that we could pay with a network, for instance, and sequence people here in Brazil, for instance. And of course, exome is less expensive. So I'd like to couple those two questions. If you could imagine something with novel sequencing data, and if it would be possible, if it's reliable, to look at the information that comes from exome, if it's possible to do some type of analysis with this type of information.
So are you talking about the COVID or any other disease?
Any other disease. Also, I would like to add the question of how much of previous knowledge do you need from the organism Homo sapiens, human data, to combine the analysis, or if you think that it's possible to do such a kind of work with plants, for instance, right? If the amount of data, systems biology data that you have for humans is so much more rich than what you would have for plants for this type of approach, or it's possible, because sometimes interfering with plants represents an answer of many thousands of billions of dollars, even if you do not change the status of the plant at all. You don't have to cure the plant, but a human you have to cure. So I would like to ask if you could use information restricted to exomes, and if you think it's possible to do similar kinds of works with other organisms that are not human.
Look, exome, if we consider what I can say, exome will give you a partial information. So,
partial, okay.
Yeah, partial information.
Too much partial, too poor information, or like 70%, 60%? What do you think?
Yeah, we can get 60% information, but when we are considering the personalized approach, it will be better for going for transcriptome approach, because from transcriptome, if I go for the whole transcriptome sequencing, from that I can identify the mutations or SNPs also.
Okay.
So
okay.
instead of exome sequencing, if I go for the whole transcriptome sequencing, okay, that will be more valuable as well as less costly. Okay, so
I know the problem with transcript approach is that you have to do triplicates or 4 samples per individual and restrict the time. But if you design an experiment where you are sequencing only control and infected person in just one time point, so it's not expensive at all, right?
Look, if you want cost cutting, then we cannot go for the what I can say, the controls for each and every analysis or every project. What we can do in one project, we can select 30 samples as a control, and that control will be for your entire lab span. Okay, so all those 30 will use for each and every project. So in those projects, we need not to sequence or find the controls. Okay, so those controls will be permanent controls for your analysis. Now,
the second question is if you can adapt the analysis for another organism that does not have so much information as human. Is it possible to do like with plants?
Yes, it is possible. It is possible, because based on the if you give me the transcriptome data and the genome data, from that we can identify each and every aspect of the particular organism. Okay, so this is possible. This is possible.
My point of view is that anything that you do with human and illness, you have to cure the person. But if you are working with agriculture, if let's use the same word, cure. If you cure only 5% of that illness in the plant, it's billions of dollars, you know?
Yeah, that's true.
With 5% efficiency. That's why I think a student should go to plants, because it's easier to cure 5% all of that illness, it represents a lot of money.
We can do that. We can do that kind of cost cutting and such kind of things. Look, if I go for the precision or the personalized health approach, for example, I don't have option for several diseases for cure. For example, if you know the Angelina Jolie's case, it is not cure. She was detected with BRCA1, BRCA2 mutations, and there is no remedy for that except this mastectomy. So that preventive measures she should take. So we have two options: either you go for the preventive measure or you go for the treatment. Okay, but both options may not be available in all cases. It depends on the specific disease or specific condition.
Sure. I was talking about cure just to do this type of comparison, because whenever you do something with plants, a partial effect gives you a response of billions of dollars. But maybe even any project adapted to plants, I think it would be very interesting to try.
Yes.
Of course, you have to do some preparations. You don't go to plants straight.
First approach should be the screening process. If I consider the human, I need to go for the first nationwide screening process. So I will know about the different markers for the specific conditions, and based on that, I can go for panel-based check. And then I can easily, with maybe within $50, I can develop the test for prediction or prevention strategy. But you need to start probably the Brazilian Genome Project, as well as specific disease-associated genome project. So in one way, you should know about what are the prevalent SNP or mutation in the entire population, and then specific disease-specific the mutations in this in this population. Otherwise, what will happen if I send a sample of a Brazilian population to MedGenome or any other company? They will use American or European marker panel, and most of the markers will be different. Maybe the BRCA1 del mutation is not prevalent or not a causal mutation in Brazil, and or maybe so. In that way, we need to first identify the nationwide or specific population specific markers or founder mutation, and based on then we can go for the cost cutting on therapy or management.
I asked you that because Vasco is leading a network for sequencing in Minas Gerais, in this state. So, of course, we cannot plan a project that's too expensive that we cannot handle, cannot pay for. But if you help thinking about an approach that could be doable, we could pay here in Minas Gerais, would be very nice to apply your approach with novel sequencing restricted to the Brazilian population. I thought that the exome would help a little bit because of the markers instead of the expression level, but your suggestion of going to transcriptome is great. So if you could think on something like that, would be very interesting, because next project for the genome network could go in this direction.
Yeah, that will be good. But at least you need to go for 500 to 1,000 sequencing minimum for nationwide.
No,
to
sure.
Identify the
I'm thinking on a network project, big one, which is not much that large amount of money, but not a short project.
Yes. No, if you invest more money, means not immediately, maybe after 2 years, if you get the fund and if you go for this kind of holistic approach, then your data will be more valuable. But because of shortage of money, if you go for example, panel-based analysis or exome sequencing, that may not be a good approach for long-term aspect, probably.
Sure. Okay. Anyone in the audience wants to ask questions? I would like to know what is dependency on bioinformatics programs, MySQL databases, whatever. What a student has to know to work for you, to work in your area?
See, the current students, they were not expert in computation. So I used their whatever computation means, simple knowledge of the handling the computer. So if you want a hardcore bioinformatics approach, the Python and R will be the most important.
Mm-hmm.
Okay, so they have to be master in that these two areas. And also, it is not that the person who is computer scientist, he should go as per his own way. He needs to take the input from the biologist, if he's not a biologist, to properly do the things, because ultimate aim will be whatever we are developing tools or methods, or it should give more than 80% precision. Currently there is no tool, means in practice it is giving above 40%, except this IBM Watson Genomics, because that is using cognitive computing. Then they are using some other AI based technologies, so that it is giving around 80% precision. So, so we need
what
to think
about
those.
What about the usage of cloud and HPC computing? Do you need big computers, large hardwares or so?
Not much requirement, not much requirement. If you have a workstation with, for example, 5 terabyte, that is fine. That is fine for developing the system, because we will have to integrate, for example, whatever we are discussing here, the precision medicine or this one, we need to integrate several databases and some analytics. So that much.
So you have you need large space in the storage. So only
not
that.
much.
You don't need much processors or
no, not required at all, because maximum thing will be through cloud or maybe with the other server. Yeah.
And as a visiting professors, can you have master students, PhD students there, or are you talking about that? How long are you going to stay?
I am staying here. Probably I'm completing
Forever.
this. Not forever, probably 4 years.
4
maximum.
years.
Yeah, maximum 4
so
years.
It's possible to have master students or to start your PhD students, and then you can supervise it from wherever you will be in 4 years.
Yeah, it can be possible.
Well, because there will be maybe some candidates for in this audience for PhD.
Yeah, I can help them if they are interested in human genomics or in the area which I am working. If they are interested, I can do. My aim is, if the students are interested for entrepreneurship development, or they want to develop some software for commercial purpose, that will be very good. So they can develop the software for commercial purpose, and they can start their own business also.
Okay.
After completion of PhD.
We are starting a program of innovation also. And the students do not need to open a company. They can work in a co-working room. We have a co-working space. You can use there. And some students can work with innovation. And whenever they have a client, the client will pay, and we will get the money, we will pay them, and we will pay you also.
That's great. That's great.
Without opening a company. So if you have some ideas like that, this kind of things are happening in India frequently. Like
incubation center. So one university, they are starting incubation center where the students can start their innovative project and get the funding from industry or start their business also. So university will support with the infrastructure, with other legal things like that. So it is common in India.
We are opening something that's even before they start initiative like that. It's like they are just students, they produce the innovation, and they get a fellowship that pays R$0, zero money, but in the month where they have the client pays and then they get the money. So is no bureaucracy. They can just start working, and we are advertising a portal, and then they can sell the service.
It's a great initiative.
Without you having the company. So if we did not start it yet, but we are getting the approval from our board of directors to approve something like that. So if you have any ideas, we could post and attract some students to work on it. And you don't have to open a company, you don't have to do nothing. Just use what we call extension program.
Just
like that. But we have contacts with people in an organization that Vasco takes place also, where you can do the spin-off, where you can do the incubation to start an enterprise as the next step, So that will be before starting the incubation, even before. Just go there, produce your stuff, we advertise, we get the clients, and we do all the processing, but the university takes 10%. That's the problem. You have to charge a little bit more because university gets some 10, 12%. You charge a little bit higher, but you don't have to pay anything. You just get the money. Right? It's a good idea. So for your innovations.
If the students wants, we can have a meeting.
Sure.
Means another different meeting, and we can discuss how it can be.
Right. Maybe we have a question from Anderson. Anderson, where are you today? Uberaba ou Uberlândia?
Uberlândia, tudo bem?
Uberlândia.
Tá me ouvindo bem, Miguel?
We are, but you have to speak in English.
Oh, okay. Hello there. How are you?
Hi, I am fine. How are you?
I'm fine. I still had not the chance to meet you personally, but I hope to do it so not too much longer. I hope so. Until the end of the year, perhaps, in my travels of the end of the year to my house, in the house of my mother. I hope so. So I have to pass through Belo Horizonte, and it's time to visit my friends.
Okay.
Deb, I have a question attached to the Miguel question. I'm supposing you are talking about a computational environment just for research. You are not talking about a production line for several people, for several patients from a hospital. It's okay. It's just for academic use that you are scaling your environment, your computational environment, because you have to store the data from the people you study, you analyze, but this kind of structure is expensive. And you say, no, you don't need too much computational resources. But I think that's not exactly like that. What can you say, please?
No, my idea is how to do a cost cutting, and in a systematic way you need to develop everything. Okay, now initially, if you suppose invest $1 billion for infrastructure, and if you don't have the customer, what you will do? So you need to develop the develop the software or develop the system for small. Then you can scale up it. Purchasing the storehouse or all those things is not a difficult job, but you should invest based on your requirement. Suppose if you are in a academic sector and you are trying to develop this one, so you keep developing and then you test whether it is giving the correct accuracy or everything. Then once you approach to a hospital that whether this hospital want to use your system or not. If they want to use your system, then you require the data storage additional capacity. So you purchase that. So in that way, step by step, you need to go. What I think. But
Okay.
initial requirement, you need to keep as minimum as possible.
Okay.
I hope it
And-
is clear.
Yeah, you already have the opportunity to apply your methodology in a large scale.
I didn't get.
Large scale.
Yes.
For many people, for a company, an hospital, or just for academic purposes.
Yes. It is not. See, always you have the ability. You should have the ability to scale up. So that window you need to keep always open. But you should not first focus on the scale up. First you develop the proper software and then scale it up. So for the software development, you need 100, for example, 100 genome, 100 transcriptome, 100 metabolome data. So for that, how much space you require, or how much computational infrastructure you would require?
Okay.
One workstation is enough for that.
Okay, and you have experience to build a computational environment for how many people, how many clients, how many patients? What?
See, in my company where I have worked, we have used mostly the software we installed in the cloud. Now, wherever you are there, no problem. You can upload your data there. Okay, from any part of the world, you can upload the data and you can get the result from there itself. So there is no direct interaction that data should send to the company and company do the analysis and will send to you. It's not like that. Everything will be automated. So you need to just provide the particular software where the people will upload their data and get their result there itself. You don't have any. We can discuss those things if you are interested, how to commercialize any idea or how to take the initiative. We can discuss in a different seminar or different meeting. We can.
You are related to some company or not?
I am.
You are related to some company, to some.
No, I worked more than 14 years in different bioinformatics and personal genomics companies, where I was in some company, I was the R&D head, where I had to develop the software or products, all those things. Means I had a team, so I had to manage those team to develop the particular product. So I know what to do or how to do. But I need the manpower. If you want to develop something, we can discuss, and I need manpower to develop this.
Okay. Thank you.
Okay.
I hope to see you someday.
Sure.
Bye-bye.
Bye.
If you want to visit us in Uberlândia, you are welcome.
Let's see. Let's see. Yeah.
Very
definitely.
dry here now. Very dry. Not a single drop of rain for several months.
Yeah.
Thank you.
Thank you so much.
Thanks a lot. Thanks, Anderson, for connecting from Uberlândia.
Yeah.
Debbie, there is a question from Maria Raquel. She is from genetics department, and she was watching in YouTube, because there are many people watching in YouTube, not in the Zoom room. So I posted here the question. She asks if are there SARS-CoV-2 reservoirs in the human body, in which organs or tissues? Are there reservoirs?
Yeah, look, as per my knowledge, I don't have the correct answer, because there are so many things going on with SARS-CoV. When you are trying to disclose something based on your research, and if it is not some journals, they don't want to publish those things, so there is a political environment. Now the particular question where it is reserved or organ or tissue in human body, so far the evidence is suggesting that this RNA can stay in human bodies, spleen, liver, and some other organs like heart also, up to 90 days, 95 days after infection. After that, what is happening, we don't know. I don't have any knowledge, but as per the publication, it can stay in liver, spleen, and even cardiac muscles, means 90 to 100 days also after infection. So they probably will be the reservoir. And that's why you are getting more damage in those particular organs, even the kidney also, on those organs as a post-COVID complications.
So COVID does not have a time like HIV-1, where the HIV-1 has a distinct targets, and then it's there in the organism, and then can pop very late on. So COVID is probably more an ongoing process, I think.
Yeah, the COVID is not over, and we have to wait to get those answers. And we have to go to get those answers. We have to monitor all those things in a long-term process. And another thing, the vaccines which we are taking, they are also creating several kinds of adverse effect, and it is not immediately expressed. It is expressing slowly. In some cases, people are getting different kinds of comorbid conditions of COVID. So that also we are trying to focus in our research. Means today I discussed with some of my colleagues if we can do something on that. So COVID is not over, and we don't know.
But for instance, I'm talking about reservoir in HIV-1, you have a person with a subtype, and later on you have the emergence of another subtype in the same person. I think it's not seen in COVID, not yet.
See, it is only
changes subtype in the same person.
HIV probably more than 20 years already, or maybe more than that. But COVID is only 2 years, and we don't know much about this. What is its actual biology, or what is going to happen? So we have to wait and watch.
Sure.
So that is the only thing.
Sure. All the isoforms are still killing a lot. You don't have an isoform that's like HIV that stays for a good while and then will affect the person very late on. This might be an adaptation of the virus. Probably it may happen. It may happen. Maybe a delayed COVID will occur. You have the virus, and then many years, then you have the disease. Because it's only 2 years maximum, and we have the virus is still adapting to the human being. Yeah. Because now, if the virus is going to adapt to our coexistence, so they are also changing its mechanism, and they may stay as a what I can say. It's not a good idea for the virus to kill the person in 5 seconds, because there could be no transmission. Right? Yeah. Virus itself, for its own existence, it should not kill its host. Yeah. So they are more intelligent than us. So they are trying to. I used to say that it's not intelligence, but it's logic. Like you have a system of tubes, and you put the water here, the water will appear in that output. So water is following logic, right? So the virus is following logic, and you don't have to have a brain. So it's a logic. It's of course okay. There's a curiosity from Santelli here. Thanks for the presentation. He says, great. And could you speak a little more about artificial intelligence used in neurobiology and GP, GA, or what's the need of artificial intelligence? What do you think? Can be applied?
Look, the terminology, artificial intelligence or cognitive computing, they're very nice terminology, but in practice, they are not so much advanced so far that it can give you the answer correctly. Because in artificial intelligence, if your if your data set, training set is not correct, or if your model is not biologically correct, whatever computation if you use, it won't give you anything. Okay, so instead of AI, if you can just use the different ML technology, that will give you more better prediction or better information. But the problem what we face, the AI guys or these computational people, they are not using much biological phenomena. So if you use biological phenomena properly, you need not to do much AI or all those stuffs. Okay, I showed you these examples of these papers. We have not used any AI or machine learning or anything. We have used simple things, but logic of the biology, how you will classify a disease or condition as a long-term consequence or as a symptoms or as a mid-term consequences. So it won't give any AI or any particular method of machine learning won't give you. You need to classify. You need to think biologically how it will be relevant and whether it will match with your expected result or the results which you are seeing, whether it is matching with that. So in that way, you first need to calibrate all those things before you develop a method or apply it. So what we did in my case, I first select the data, and you have to select data which is giving you the maximum precision. Now, in these 5 different data sets I have used, I have also used other 10 different data sets, but they are not giving me any good result. They are giving me maybe 50% precision using the same method. That means the data is having some problem. So you need to standardize the data. So first, source of data and standardization, that is important. And then if you use whatever in the method, it will work. But data and those analysis will be, or standardization of data is very important from the biological point of view.
Okay.
Okay.
All right. Sandeep, are you around there? I think that it's time to thank Dr. Debbie for this great afternoon. It was great to listen to you and have all those discussions. So thanks a lot. It was a nice opportunity for us and for the students too.
Thank you so much for joining, and I hope I didn't bore you people with my with my accent and with my.
And you know, in the web link that we have in the BrazWebinars website and also in the PhD program by Informatics, we have the YouTube taped also. So if you like to share with other students, for the people that are taking the workshop. So, right, Sandeep, you can use this talk also for an introduction to them, okay?
Yeah, that could be.
Sandeep, it's with you.
Okay, so thanks a lot to Professor Dev for such a nice talk. It was really interesting, and I hope everyone likes it, and will contact in future. And also, again, as a reminder, the discipline is starting on this area from 12th to 16th. Yeah, 12th to 16th. So if someone is interested, they can contact the Bioinformatics department and they can know a little bit more about it. So thanks a lot, Professor Dev, and thanks a lot, Professor McGill, and all. Thanks a lot. Thank you.
Thank
Thank
you.
you.
Thank you, everybody. Bye-bye. Ciao.
Bye bye. Ciao, people.
Ciao. Have a
See you.
nice weekend. Take care.